My name's Anthony Richard Monte Carlo III, and this is my project on investigating G-Quadruplex mimicked enzyme kinetics for molecular sensing. I hope you enjoy. The COVID-19 pandemic has made it abundantly clear the need for accurate and rapid diagnostic tests in order to shorten the turnaround time for someone to obtain their results. One such way that we can improve the turnaround time is by utilizing point-of-care tests. Point-of-care tests also aim to lower the cost-prohibitive barrier to accurate diagnostics and provide platforms which are quick, accurate, and easy to use in order to improve the turnaround time. Point-of-care tests require two different components, one being a method of detection and one being a method of signal generation. Signal generation is the focus of our research and is important because it tells us whether or not the test comes back positive or negative. One such way to generate signal is to use an indicator species in an oxidation reduction reaction in order to produce an output. That output could be in the form of chemiluminescence, fluorescence, or in our research, color change. A commonly used indicator species is ABTS, which when oxidized shifts its absorbance spectra to 420 nanometers in the blue light range. The more intense the blue color, the more we can say the reaction is proceeding. What's interesting about ABTS though is it has multiple oxidation states, whereas the overoxidized state does not have such an absorbance and can contribute to signal loss. In order to catalyze this reaction, we also need a peroxidase enzyme, commonly in the form of peroxidases like microperoxidase and horseradish peroxidase. The problem with using enzymes, though, is that they can be easily denatured by extreme pH conditions or different temperature conditions, which can lead to a malfunctioning test. G-Quadruplex hemin is a DNAzyme, which can mimic the behavior of these peroxidases. A DNAzyme is comprised of a functional cofactor, in this case hemin, and a G-quadruplex, or the DNA part of the DNAzyme. G-quadruplexes are guanine-rich DNA strands which can form multiple different structures depending on the sequence. When G-quadruplex and hemin hybridize with one another, hemin shows a very enhanced peroxidase activity, which can be useful in point-of-care tests. In order to further document the potential for use in point-of-care tests, we set out to investigate the impact of pH condition and buffer type on the behavior of G-quadruplex hemin. Moving into the methodology and analysis section, we see in Figure 1A and B the raw reaction curve of G-quadruplex hemin across different pH conditions and in different buffers. Interestingly, we see that there is a signal loss, specifically in Figure 1A, under basic conditions. We find this interesting as, again, we mentioned that ABTS has multiple oxidation states, and so further experiments plan to document whether overoxidation or reduction is happening in these systems and is contributing to signal loss. We also see a more stable signal in Figure 1B, and so we believe that buffer type also has an impact on the amount of signal loss. Also of interest to us in Figures 1C and D is the amount of enzyme death on the loss of signal. As we see in Figure 1C, we see a sharp increase in signal followed by a general plateau. This is further emphasized in Figure 1D. We believe that hemin exposure to the overall environment may also contribute to the death of the enzyme. And in Figure 2, we show a comparison of the rates, or initial velocities, of these different enzymes across different pH conditions. And so in general, we see that G-quadruplex hemin shows reduced kinetic activity in PBS buffer versus TBS buffer, shown by the lower initial velocity and lower peak signal. And in basic conditions, signal is degraded faster in TBS buffer versus PBS buffer in G-quadruplex hemin. We hope to further investigate this using different indicator species such as Amplex Red and TMB, as well as really teasing apart the impact of enzyme death on the loss of signal. And with all this, I would like to acknowledge the Rutgers Camden MARC program, as well as the NJSGC for funding this research. I would also like to thank the Army Research Office, PCASE, and my fellow lab members for all their support throughout this semester. Thank you, and I hope you enjoyed.